Thanks so much to the organizers for having me. It's an honor for you guys to give me a bit of your time today. So, yeah, I wanted to speak to you a bit today about how generative AI is impacting tasks in drug discovery. And I realize that the title of the talk is a bit measured when I say tasks. I'm not talking about the whole industry being completely disrupted like the taxi industry was by Uber. So in this case, I think what we are doing is making inroads in the hundreds or thousands of tasks that make up R&D. So I want to show you a few vignettes to kind of construct a narrative on how things are changing, how we may be getting more efficient and more creative. But when I was putting the talk together, I was curious, when did we start using these approaches, machine learning? And I've been at Novartis for 23 years doing research. And as far as I could find, the earliest paper we published was back in 2004, where we actually used the term machine learning. And we were basically docking compounds into protein pockets and then building QSAR models on the docking hits that were active. And then that turned out to be better at enriching for true actives. So that's to say we've been doing machine learning for over 2 decades in the pharma industry. And then it struck me as curious, we've never actually said we're making machine learning drugs, but I keep hearing this question, when will we have our first AI drug? And it's odd, we never talked about having electricity drugs when electricity came in. So I actually just think this is not an interesting question. We shouldn't be asking when are we going to have an AI drug, because it's sort of meaningless. We're using AI across the value chain. And the goal would be to make us more efficient and more creative in what we do. And I do think deep neural networks and generative methods are addressing these tasks. And if you just think about the entire R&D continuum, it's roughly half research, half development, average timeline 12 to 13 years. How can we shrink these pie slices? So that's what I'm interested in, is getting a little faster and better at what we do. I've put some asterisks next to 3 things I want to talk about today, which is target ID, assay development, and hit finding. So these are areas I'm passionate about. So before we get there, I just wanted to disambiguate a little bit machine learning in the sense of discriminative, which is the classic form where we're sort of predicting a label or a class on a sample. And this could be like, is this compound active against this target? Yes or no, give me a score. So the real shift today is this generative AI, where we're actually understanding underlying patterns in input data in terms of probability distributions. And using encoding methods into a latent space and decoding those, we can generate new samples from that data. And so that's where it gets interesting, that AI is actually suggesting molecules, suggesting protein sequences. And these are just some examples of generative AI. You're all familiar with ChatGPT and language prediction, where we're learning basically how words attend to other words in the context of sentences so that you can do language prediction. The sky is starts a sentence, blue may be the most likely next word. But this doesn't stop with large language models. We can do things like image to image translation, or even sample from chemical space. So I like this figure. This is not my own figure, but here's an example of taking a reference molecule that's active against a target, and at the far right is another molecule that's active, and just sampling the latent space in between those two. It becomes an interesting way to find more actives or even potentially hybridize those 2 active molecules. So this is where generative AI is playing. So target ID and small molecule design and assays are very early parts of the pipeline where we're applying generative methods. Now, importantly, there's a lot more to R&D than these 3 steps. And I'm not obviously covering biologics design, PK/PD predictions, retrosynthesis. So there's just tons of places where AI is being applied in drug discovery. So target ID. This isn't obviously a super critical part of drug discovery, because if you pick the wrong target, you're going to waste a lot of time and money downstream. So we really want to put our best foot forward on picking the targets that we're going to develop therapeutics for. So classically, we would do things like understanding tissue expression, applying functional genomics like CRISPR or RNAi approaches to understand what knocking out this thing does in a phenotypic setting. And then, of course, human genetics with all the emerging biobanks and sort of massive GWAS studies out there, we can get an early hint of how gene variation relates to having a disease phenotype. And this is obviously a powerful way of getting at targets from human data in the wild. And then large language models. I mean, of course, we have Claude, ChatGPT, all these available to us, but there are also companies that are making more science graphs that have prompting and LLMs on top that can really get deep access into the literature quickly to support your target selection. And then there's a newer field that's a few years old now where people are building single cell foundation models across all of the single cell atlas data that's in the world and using this to simulate perturbations like you would do functional genomics, except we're simulating gene knockdown and knockup, and I'll talk a bit more about this. But I like to use this analogy for this part because you might be shown this picture and asked, what is unusual about the interactions of the people at this party? And the first thing that I would think is, I don't really know, because I don't know who these people normally associate with. And so then the question would be, if you had data for these same people from 100 million previous parties, could you tell us what's unusual about this party? So this suggests, given enough data, you could generate a pre-trained model that could then suggest what's wrong in a certain context. And of course, we can borrow from the transformer field, the language field, where we understand how words attend to other words in sentences. And I'll just extend that analogy to where instead of words, we're talking about genes, and instead of sentences, we're talking about cells. So in a similar vein, not exactly the same, genes do attend to other genes in the context of cells. So we can pre-train a transformer across single cell RNA-seq data from the human body. And this is not my idea. I'm presenting other people's concepts here, but we're using them. And with these pre-trained transformers, we can fine-tune them on a disease versus healthy setting. So maybe everyone's using the same pre-trained transformer, but you have your own proprietary single-cell RNA-seq data for disease versus healthy in a tissue. So the model gets fine-tuned on that, and the weights learn the difference between a healthy and diseased cells. And so these kinds of transformers can be used for many downstream tasks, one of which is just cluster the cells. So it wasn't trained for that task, but actually these models do a really good job of separating cells by type based on the genes they're expressing. You can also construct gene regulatory networks from the attention maps in these models. And then finally, this concept of in silico perturbation. Can we actually, now that we understand how genes are talking to other genes, what happens if we knock out a gene? What's the effect on the others, right? Just like knocking a word out of a sentence. So we've been doing this on many, many diseases, and just to say that sometimes these in silico perturbations actually do predict what would go on in terms of like differential gene expression. This is from Christina Theodoris's Gene Former paper. This is one of the original types of these foundation models, and she showed that knocking down these transcription factors, GATA4 and TBX5, doesn't show much of an effect on housekeeping genes in the cell in her simulation, but it actually does have a profound effect on known target genes of these transcription factors. And that was just a way of showing that there is biological meaning captured in these in silico perturbations. We published this paper, and there's another one called SCGPT, which you can see the trends are roughly correct in terms of gene directionality when you do these in silico perturbation, but the magnitudes aren't always right. There seems to be a hot trend now for people to bash these sort of models and say actually baseline methods work almost as well at predicting gene expression, but I actually do believe, based on empirical results, that these methods are pointing to some biology relevant to disease when we actually go to validate targets in the lab. So I think the jury's still out on whether these things are are working as advertised. And so why would you even want to simulate perturbation data as opposed to really doing it? I mean, you can knock genes out, you can knock them down, you can knock them up, and this can be done genome-wide, purely in silico, really on all cells and all cell types found in a tissue. So you simply can't do CRISPR on tissue like post-mortem tissue. So you can simulate this. And CRISPR screens coupled to sequencing like perturb-seq or crop-seq, that's the closest sort of lab analogy to what we're doing, what we're simulating here. They're great, but they're expensive and limited to really cells that are amenable to that sort of protocol. So while I like this approach, a lot of cautionary tales here. Healthy single cell RNA-seq, which the models are largely trained on today, don't really understand all possible perturbation states. So healthy cells don't see what it's like when lots of genes are knocked down. So the training data doesn't contain all the information it probably needs. And actual perturbation data like perturb-seq data is still super limited in the world. There's a lot of proposals out there for consortiums to build this kind of perturbation atlases, which would give us a real better training set for making like perturbation models. So another problem is anytime you do a computational method, a bioinformatic method, you get gene lists, right? So what do you do with gene lists? If you're a computational biologist, usually you put these into set enrichment algorithms and predict pathways and processes that are reflected in your gene list, right? But the interesting thing today is these large language models. So one company working on this is Causally. So they have a Gen AI platform that has millions and millions of biological relationships captured both from published literature as well as clinical trials, GWAS catalog. We have well over 3,000 users and research using this, which is, 60% of our research organization. And these kinds of things can generate really fast AI reports based on your user prompt. And I'll just show an example. So I put this in, I want a list of novel potential drug targets for malaria targeting the plasmodium, the parasite. These targets could be essential at different life cycle stages of the parasite. So enter this into Causally. It essentially launches an agentic type of search that ultimately took 1 minute and 26 seconds. In the end, it referenced 32 different documents that it found that were highly related to the question. The agentic deep research aspect actually reprompted my question with 60 individual different searches to really accurately answer the question. And then it output a 10-page report with 29 different target classes. Many of these genes it suggested were uncharacterized but essential genes to plasmodium, and they were predicted to target different aspects of the parasite's life cycle. And so, that's to say it would have taken me easily a week, maybe 2 weeks to compile this 10-page report by reading these 32 papers, but this was 1 minute and 26 seconds. So this is the power of putting such gene lists you may be getting from other AI methods into an LLM to quickly annotate and provide more evidence towards a target hypothesis. So, not showing any targets or disease names here, but this is just kind of how we work. We'll do these in silico perturbations using healthy versus diseased tissues. We'll layer on genetics and genomics if we have it. And then we'll use things like Causally or even open targets to further score or annotate gene lists and come up with gene candidates. And we have one example of using an organoid disease model. And the nice thing about organoids is they're closer to human biology than a simple cell culture. However, you can't necessarily test every gene in the genome in an organoid. It's super expensive and time consuming. So that's the power of AI here, is we can actually suggest a much smaller number of things to test that are already filtered with prior knowledge. And we tried some compound screens for these targets as well as a bit of CRISPR. And a couple of these things are entering mouse experiments. So I hope this is the kind of workflow that not only makes us, I don't know that it makes us more efficient per se, but I think it gives us access to hypotheses we may not have gotten otherwise. So AI approaches are accessing novel target spaces, and combined with genetics and LLMs, we just have a stronger evidence package. What's the bottleneck? Obviously, it's still experimental model systems. If you don't have a good model system to test your AI hypotheses, it doesn't matter. Even if you get a hit in your primary assay, it's going to take a year to really validate that target, or 2 years if it's novel, and we need to understand the biology. So as far as I'm concerned, there aren't many shortcuts today using AI to do target validation. So, we solve one thing and the bottleneck shifts elsewhere, and almost always it shifts back to the lab. Okay, so say we have a target we like. Now we can move on to designing a molecule. I'm going to stick with small molecules because that's my space more than biologics. What is the problem with finding compounds for drugs? The fact is there's sort of a lot of parameters that need to be optimized in place for a compound to become a drug. And I would just say often a drug candidate, it can't just be identified, but we need to maybe design it to fit the right properties we need also for the shape and electrostatics of the target pocket you're going after. So there's too many molecules in the universe. We need to be more rational about how we select from that chemical universe. And I think to do that, there's, in my head, three challenges. The first is generating ligands using generative methods. And of course, you can screen pre-existing molecules, but maybe you want to actually design molecules out of the gate that are better for your target. And they need to be synthesizable, of course, with good properties. The second is kind of understanding how the compound and target are binding in a pose, or these days we might say a cofold. And then to predict the binding affinity. So we can design a lot of things, but if we can't rank them using energies or binding energies, then we don't know what to make first, right? So these are the three problems. And AI is actually addressing all three. So the ligand design piece, this has been going on for, I don't know, over a decade now, where we can use molecules and encode them into a latent space and then decode them, so generate a sample. And you can even do some multi-objective optimization in this process where you're only picking in the latent space that has the properties that you want for the molecule. So you can take non-optimal molecules and then find improved molecules in the area. So that's generative chemistry. And immediately I was interested in this idea of 3D generative chemistry. So why don't we just design molecules that are conditioned on the pocket that the molecule's going to bind in? So just grow the molecule in the pocket. Since 2020, there have been these 3D Gen Chem methods. The initial ones kind of had low drug likeness, so these got better as people improved these models. But around 2022, we picked up this academic algorithm called pocket to mole. I just want to call out how important open source academic algorithms actually are to real industry and drug discovery, because these are the algorithms that people can immediately test and they actually start to launch the field. So, hats off to academics that are putting these things out there. Not to say they shouldn't try to monetize at some point with a company, but this is how things get started. So we took this pocket crafter, we took pocket to mole and we built it into just a pipeline to make it easy for our scientists to use. This pocket to mole starts with these sort of frontier atoms, and then it tries to sample around a focal atom and creates the next atom and bond that would be relevant to the pocket shape. So it's a little bit like Jenga, or I don't know, it's Candy Crush or something. Just can we fit the molecule in the pocket with the shape and the electrostatics and all that? So this is a typical pipeline, and you can still generate a lot of molecules that could fit in that pocket. So we might make a 500,000 compound diversity set de novo, where we think it could fit in the pocket, and then we'll apply further properties, drug-like properties. We'll even rescore it with a different scoring function, and then propose it for de novo synthesis. And the nice thing about 3D Gen Chem is it gives you the predicted pose, so you can see what are the actual interaction sites on the protein for the molecule you're designing. This is just one real example we published. We were looking at MYC, the classic oncology target, and one of its interacting proteins, WDR5. And we asked, can we inhibit the binding interaction between WDR5 and MYC? And so we had a sense of a pocket on WDR5 where we could design molecules. We did go ahead and screen 1.1 million compounds against this protein-protein interaction. And obviously, you see we had a number of hits at 40 micromolar cutoff. Using Pocket Crafter, we suggested a couple thousand molecules. Obviously, fewer hits, 7, but the enrichment rate here is like 13-fold better for the computational method over sort of a background random screening. So it suggests this is possible. And these are a few of the molecules. They're analogs, but they are low micromolar activity in this PPI assay. They don't hit the counter screen. And, the important thing here was we actually knew the residues in WDR5 that the molecule was interacting with, and we think that it was accessing residues that were not bound by other hits that we found through screening. So that's the power of sort of de novo design to sort of get what you want up front. Okay, you can design molecules, but also you might want to better understand how they're binding your protein, especially if you didn't use a 3D Gen Chem method. So these are just a sampling of some of the co-folding methods that are out there in the world. Many of these are open source. AlphaFold 3 is still open source to academics, not commercial. But this has really been a game changer because unlike classical docking where you have a static protein structure and try to fit a molecule here you're literally co-folding the protein in the presence of a compound. So you're kind of getting that dynamic, flexible aspect in the binding prediction. Most of these put out a confidence score, which isn't actually a binding affinity. Boltz 2 is kind of an exception. It has a affinity prediction, and that gets us towards how do we actually rank designs from generative methods. So there's a field that's really exploded. Some people call it affinity models, other people call it drug target interaction, DTI, but there's a ton of methods every week coming out where people try to predict the KD of the interaction of a compound to a target. And it's very interesting because you can represent proteins in a million different ways, starting from just its sequence, but also to secondary and tertiary features. And same with compounds. Compounds can be represented many ways. So what people are doing is creating these joint models that learn on protein features and chemical features in their sort of statistical correlation, such that you give it a protein and a compound and it tries to predict the affinity. One of the methods a postdoc of ours worked on is called BALM. You can see the Pearson correlation of its predicted affinities to real affinities is somewhere between 0.5 and 0.6. That is better than just classic docking scores. But I do want to say it's not near the best-in-class methods, which are still physics-based, free energy perturbation. These numbers would be closer to 0.8 Pearson correlation. So the real challenge here is how do we use these AI models, which are super fast to use, to be as accurate as our slow and computationally expensive physics methods. And maybe it's some hybrid of AI and physics approaches that will get us there. So some takeaways on that is, it really is, Gen Chem is accelerating access to chemical space with better properties and scaffolds that are target specific, but ranking those designs at scale remains a challenge. So we need to be better at ranking. Where's the bottleneck now? Well, it's back to the lab, it's synthesis. So, we know how to kind of use automated chemistry to make families of molecules, 1-2 step reactions, but the future, if we really want to do a lot of de novo design, is going to be more autonomous chemistry platforms that are multi-step and can really make diverse singleton type molecules in a more autonomous way. So I think that's what the world is waiting for. Okay, so the third venue is around AI-enabled assays. So I'm excited about this one. This is back to kind of imaging, which is sort of the root of a lot of these deep neural network methods. We use a lot of high content screening in pharma as assays. So we can do automated microscopy on these high content imagers and get such cellular images like this, where different features of the cell have been stained with dyes or fluorescent antibodies. And that's used a lot for phenotypic screening, understanding the mechanism of action of your compound, safety, etc. But it is labor intensive, and fixing your cells actually fixes them in time. So once you've taken that image, you're kind of done with that cell because you fixed it and stained it. So that's a problem that we're addressing here. So while the top workflow is purely kind of an in vitro workflow from molecule to cell to stain cell to imaging, we've been working on this in silico labeling approach where we have a molecule that we image using bright field microscopy and we don't stain, and we try to predict the fluorescent image from the bright field image. So one has to obviously train models that understand both bright field and fluorescent images. And we're borrowing from, again, public algorithms. This one's called Pix2Pix, and it's like a method that was published to colorize black and white photos, which is essentially the same task we're going to be doing with cells and bright field images. So we literally applied this Pix2Pix algorithm in the context of training where we paired a bright field microscopy image of cells with a fluorescent image. And we did this for multiple sort of channels with different stains. This is kind of reflecting the different stains you might see in the cell painting protocol from the Broad. And then with that pre-trained model, we can take a bright field image as input and use the various different models that understand stains to predict the fluorescent output, and then you can merge them, and then you essentially get an image that looks like you had done a cell painting protocol. So one of the ways we used this very early on was a myelination assay. So myelin is this protective sheath that's wrapped around Schwann cells and protects the neurons. And we're interested in, in some diseases, you lose this myelin sheath, they get demyelinated, and we're interested in drugs that can actually regrow this myelin or increase it or protect it. So we developed an assay where we could take a bright field image, we could do the staining, that's the top right image. This particular antibody we had wasn't great, it crashes out of solution, you get a lot of, punctate stuff that's ugly. But we sort of trained a model around that. We also trained it to not predict neurofilaments, which is not what we're trying to see. So the bottom left would be like a false positive. It's not what we're imaging. So you can see the prediction on the bottom right for a model that's been trained on both bright field and myelin stains. I actually think it looks cleaner in most by eye than the actual stained images. And our Pearson correlation was like 0.83 between the actual myelin area on our Yokogawa microscope in the predicted area using this model. So, shifting to bright field on this project we were working on, it enabled non-destructive kinetic reads. So you could take a bright field image, wait a day, take another one. You actually get a sense of what's going on with myelin over time, which you would not have gotten if you'd fixed your cells and stained them. The reagent requirements were reduced by like 80% because we didn't need the fluorescent antibody anymore. And we got 10 times faster in actually doing this imaging on the project. So, and these kinds of approaches are scalable to multiplex screening. So this was just myelin stain, but you could throw this through any kind of pre-trained model. So it's really transforming the way we do imaging when the model can see enough in the bright field image. How else can we use this? Another problem we face is we've got hits in an assay or against a target, and we want to expand on those hits. So this is sort of a hit expansion problem. Classically, if you've got an active molecule, you might just find analogs of that molecule in your library and test them. But there's a lot of other more interesting ways of doing hit expansion. So this is an attempt to create these in silico labeling profiles for a large number of compounds. And then we could use a molecule of interest and query all of these in silico labeling fingerprints for compounds that could have similar biological activities. And did a little bit of benchmarking. So across 279 legacy assays we had, if we use this hit expansion approach using true stains, we get something like a threefold enrichment for new hits. ISL is essentially identical. So the purely virtual version can recall just as many new actives from the archive as actually doing the imaging. And Morgan is a type of 2D chemical fingerprint, so you can see the hit enrichment is not as good when you just search for analogs of a starting molecule. So now that you've seen in silico labeling, I want to shift to something that I think is a bit more science fiction-y. This is the bottom row. It's a method we call PDIF. And so the question we had was, could we start from a compound, generate some sort of feature representation of that molecule, and from that create a microscopy image? And you can see that there's only red arrows in this flow. So this is a totally in silico experiment. Can we take a molecule and predict an image that it's going to have if it were applied to a cell? So I thought there's no way the team's going to pull this off because there's just not enough data in the world of cellular images and chemical structures to train a generalizable model. So, this is like generating images from prompts in Stable Diffusion. You can ask to draw an astronaut riding a horse, but can we do something like create me a high content image of U2OS cells treated with a CHEK1 inhibitor? So this is a sort of strange request that we're going to make of this model. And I mean, one of the keys here was really how we represent the compound in this model. So we can have a real or a virtual compound, and we need some way of representing it. In the classic way, everyone would use as a 2D chemical structure fingerprint, but we did something different. We used what we call an in silico bioactivity profile for the molecule. And so I'll take a bit of time to explain this. This fingerprint is based on more than 10,000 legacy dose response assays that we've run over the last 2 decades at Novartis. This is across cell types, protein types, disease areas. It's basically all of our dose response data. So whatever, it's probably $1 billion worth of data. You can build a QSAR model on every single assay we've ever run, right? And then you can run a compound, real or not, through this model and predict its activity in every assay we ever run. We call that profile QSAR. So it's this sort of biological fingerprint. And I think the power of it is that it really doesn't treat all atoms and bonds the same in a molecule. It actually emphasizes pharmacophoric type of motifs that are biased for pharmacological activity. So I think it has more power than a typical chemical descriptor. So we did use this PQSAR biological profile to train a model of paired molecules to images. And what we did is we trained the model using nearly 4,000 compounds that we know have biological activity in something we call the MOA box, which is screened over and over on our phenotypic assays. And we profiled it in U2OS and generated cell painting data for those compounds. And then we computed the 2D fingerprints and PQSAR profiles for those same 3,750 compounds. And so, we did sort of a cluster-based holdout test, 90% of the data for training, 10% for testing, as you might. And here's some of the results we've published. So just for 3 of the molecules, you can see the real cell painting image on the left. And with the model we trained on chemical fingerprints in the middle, you see basically total nonsense and hallucination. This looks nothing like the actual image for that molecule. But using this biological activity fingerprint, we actually got pretty good looking images that looked like the real cell painting images. So this was a surprise that this worked. And we thought, all right, how can we use this? Why does it matter? You can predict an image for a compound. So this is back again to hit expansion, which is always a problem on how do we take a molecule and find more things like it. So in this case, we could have a molecule of interest that has a certain cellular phenotype. Can we just find other molecules that might have that same cellular phenotype purely in silico? And so we did this. We tried retrieving top 50 molecules with real images, top 50 molecules based on simulated images. also with 2D fingerprints. And so at random, the overlap of these two sets would be, up 10, 12%. With 2D fingerprints, it's also quite small. Using the biological fingerprint itself, actually the overlap is closer to 40%. But this PDIF method, which is the method I just described, where we simulate an image, actually has about a 50% overlap. So this suggests that this PDIF can be used to retrieve more molecules that are active, like a query compound, literally without doing any experiments. So the takeaways, in silico, labeling massively saves time on cost while enabling kinetic time point readouts, not destroying the cells. These generative images can be used for hit expansion to find more active molecules. What's the bottleneck? So the question is, do we need new in silico labeling models every time we switch a cell type or a stain? Today we are building a new model every time there's a new stain. I think we'll have to, but we are trying to see if we train a model like on one type of neuron, is it transferable to a slightly different, but also another type of neuron, so that we save on all this model training. All right, so to wrap up, I'm kind of interested in this question we get asked all the time: what is AI doing for us? I think obviously this is the high level goal. We have traditional drug discovery. The ambition would be some kind of increase in efficiency. Can we get faster what we're doing? Can we make fewer molecules? Can we test fewer targets? But also, the x-axis is a bit more like a first-in-class axis. Can we get access to new hypotheses that we wouldn't have gotten without AI? I think maybe we got here back in the days in the 80s and 90s of combinatorial libraries, where we could actually screen a lot of molecules, but they were all kind of really similar because of the chemistry. And so you weren't necessarily increasing access to new hypotheses as much as efficiency. So just throwing CombiChem under the bus a little bit. But so I think today we're more like here. This is just my view. I think we are getting more creative, and we're coming up with target hypotheses that we wouldn't have gotten otherwise, molecules we wouldn't have gotten from screening existing molecules. So the creativity is up. It's going to be difficult to rapidly change how efficient drug discovery is, partly because it's so many hundreds or thousands of tasks over 12 to 13 years. But that would be the ambition. Can we make fewer molecules, test fewer targets, to make the whole thing faster? I think we can get there with method maturity, access to methods. Success will build trust and adoption. I also think a bit of this shift could come from autonomous labs and more lab robotics. So I don't think it's all going to come from AI. As you could see, a lot of the times the bottleneck shifted from data analysis back to the lab being the bottleneck. So I think what we're really going to need to hit this AI ambition is a combination of lab automation and AI working together. Yeah, and with that, I'll just end with a quote that I like from David Brooks that said, "AI will reveal who we are by what it can't do." So I'm very interested to see where that limit is on AI, and lots of people to think, and appreciate your time and listening. Thanks.
All right, questions?
Hi, you talked about pockets, and I was wondering if you can touch base on disordered molecules. On your last point, what AI can't do, cannot predict those disordered regions, and how do you deal with those?
Great question. I think for disordered proteins, I believe most of the time they are ordered in some scenario. So part of the trick will be, can we use AI to predict a binding partner that orders a disordered protein? And then that's the context when you would want to do co-folding with small molecules and target a more structured complex. But I wouldn't take a disordered protein and try to find a docking pose for a molecule because I think it's just missing whatever the inherent cellular context probably is. if it's a transcription factor, it probably needs DNA to be, or a binding partner to be more ordered. So you have to kind of maybe use AI to pan through different potential binding partners before even thinking about small molecules.
All right, in the back.
Thanks very much for this great talk. My first question is about your partnership with Causally. So you use their platform to improve target identification, let's say, but do you go back to them with like, oh, we tested this molecule in the lab so that they can refine sort of their solution space? Because that's where these kind of companies don't have the scale to like test things the way that big pharma would. And then the other question would be for small molecule development, right? And for antibody development, there are always like these really slow, very expensive, time-consuming steps. If you had to like go to a chemist or go to a small biotech and be like, these are the things that are causing me misery in the lab, like, could you name a few things that people could maybe really focus on method development to make it cheaper, more scalable, so that you can collect data about those rate limiting steps more readily? Thanks.
Cool. Yeah, a couple very different questions. Let's start with Causally. We certainly partner heavily with them on feedback about the tool. I haven't really considered that we go and test something and feed that back into the knowledge graph. Their system it's called Causally because it does try to understand actual relationships, like positive and negative relationships in literature to get that causality. But today I think it would be a drop in the bucket if I gave them my data compared to all the data in PubMed. So I don't know how much it would alter the science graph, but it's an interesting idea. Bottlenecks in the lab for chemistry. I mean, I'm not a chemist myself, but I think things like automating purification, automating more multi-step synthesis are bottlenecks. What else? I mean, we just need to get faster at making singletons. That would be my dream as an AI person. But it's a great question. I think there is a huge role for academia to play in all these advancements, as there always is, Okay.
All right, right here.
Yeah, fascinating talk. Thank you. So my question is on the small molecule in silico design. How far along are we in the toxicity prediction and preclinical tox or even patient safety?
Yeah.
On that side.
Yeah, I think it's not far along, is my opinion, speaking for myself. I think it's a data paucity issue. I think we just, even if we compile data across all pharma and biotech in this tox space, is just very difficult because of the number of mechanisms, the size of chemical and biologic space. It's so much harder to predict tox and the many types of tox. We're trying. I think everybody is.
Yeah.
I would say, ADME is much better. Things where we can measure, of course. Yeah. But maybe the right way is really just to take it one at a time, like just cardiotox, just liver tox, and try to build a lot of data sets around, a proxy assay for that. And that's as good as we can get at the moment. But I would say we're definitely not able to replace actual tox studies yet.
Okay, thank you.
Yeah.
Yeah, my second question is actually the follow-up of the Causally. So have you compared the effectiveness or accuracy of Causally to some of your in-house build based on your graph, so data foundation? Have you compared that?
Other types of LLMs, do you mean?
Or based on your own data product, or
I see.
Yeah.
Knowledge
graph.
So it is built on what's declared in the literature. So it's really only as good as science is out there in the world.
I see.
I think the best it can do is understand when there's conflicting information, and it does at least attempt to understand if evidence is controversial or strong. So it does give you a ranking for its assertions, like a score.
I see.
And I haven't tried to put it head to head with actual data that we have to see, but I think people for years have said a lot of what's in literature doesn't validate when we try to do it in-house, but there's so many caveats to that. But that's going to be the same problem when you build an LLM on literature.
Yeah, thank you.
Yeah.
Hi, you mentioned that physics-based models for calculating binding affinities are still better than the AI-based models. What do you think is needed to get the AI models on par with that?
Yeah, I mean, I think this again could come down to more KD data across more target classes. I think it's just more data. And most people are training on the same publicly available KD data sets. We actually have a lot more KD data in-house than what's actually in public databases. So we're seeing, can we combine internal, external binding affinities to make an even better model? But then you're just going to be limited to the diversity of targets that have been drugged or liganded before. Whereas physics and theory, you could start with a protein with, that's never been drugged and make a prediction. So more data is usually the right answer.
Makes sense. Thank you.
I'm thinking about a meta AI because you did a lot of things and was wonderful. You put together almost like a multiplex integrated from different area of expertise, but in the drug discovery and particularly, for instance, like you pick a simple case like a pancreatic cancer. Okay. So you cannot use the, classical traditional AI or even with the agentic AI, you only give you part of the pictures. We need after this, we call the meta AI, and you build another higher dimensional model. I call it the network analysis with weight. So you can correlate, for instance, like, look into the pancreatic cancer, the intrinsic and the extrinsic environment, the parenchyma, transformative, and also, for instance, like the BRCA1, BRCA2 also kick in place on top of the mutations, you're talking about. And if you lost that kind of insight, well, you get wonderful a heat expansion molecule, but you forgot that, how about the immune environment, and others, molecule, they may interact or they related to each other. By how much? See, 0.85% or 0.42%. So I call this meta AI network analysis across those modalities.
Yeah, so, I certainly didn't thanks for the question. I didn't get very far into like multimodal or multi-omic data, which would be required for representing tissues and more holistically organisms. So I think, but that is a field of putting together genomics, proteomics, genetics, metabolomics into joint models, which are likely to be more active. It's not always easy to get that rich of data for the same kind of samples, but I think in something like pancreatic cancer, it's obviously there's going to be cell autonomous genes, but also crosstalk in the microtumor environment. So, this is why people talk about virtual cells and then virtual humans, because everyone knows we need to model the complexity at a deeper and deeper layer. And it's going to be a matter of building those data sets as we go. Yeah.
Well, thank you. At least we're on the right track.
Yeah, exactly.
A great talk. So this is a question originating from a biotech perspective. If you had a choice to buy data or a model or a combination, what would you choose?
That's a great question. If I had to choose, I would say the data, because I think obviously you could then put your own flavor on models. I think we're pretty good at building AI models, but data is really the gold nugget underneath. And I think having that, then you could do different types of stuff with it.
Okay, thank you.
Yep.
Makes sense.
All right, any other questions? Yep.
Excuse me. Have you made a library or kind of generated a list of all those molecules that you prepare through that pocket profiler or whatever you called it? I forget the name already. And then done predictions on those things, on those molecules?
For the example I showed with like MIC and WR5, or do you mean more generally?
More generally.
Yeah, I mean, I think we can generate these huge lists using GenChem approaches. I don't know that we even necessarily have to keep those, but we will take them and then run predictions on them, so further refine to pick and then make a few. But you can generate a vast amount, and I'm not sure it's worth keeping them all, but we'd certainly keep the filtered lists that we make predictions on. What kind of philosophy?
In the sense, are different types of parameters that would you use for fitting into the pocket, correct?
Yeah.
So there should be some kinds of principles you'd apply for how I want to design those pockets and what pockets am I looking at?
Yeah. Well, I mean, the 3D generative methods, the actual design will be conditioned on the properties of the pocket. So it'll be electrostatics, hydrophilic shape, and those are sort of built into how it would grow the atoms and bonds as it generated it. Oh, we can follow up later if I'm missing it.
So I was interested in the image generation part, and it does seem like science fiction that you can go from molecule to image. Is there a worry that you're the data is obviously trained on cells that are easy to image, so then it would maybe be biased in that direction? So adherent cells being the hallmark there.
Yeah.
Is there a chance to kind of combat that by moving into like more non-adherent assay types like flow cytometry? I mean, once you've got the images down, like it feels like the sky's the limit. You could start thinking of just about any assay. Is that a direction that you guys are looking at?
Yeah, that's super interesting. I mean, to your first point, yes, for sure. Everyone uses U2OS because they're big and beautiful. So we've tried this in other cell types. It's U2OS is the most performant for something like cell painting. But so there's definitely, maybe we're over optimistic on how we would do in other cell types. I like that idea of using this in flow cytometry. That's pretty cool. If you can compute these fast enough, great idea. Not trying it yet. Yeah.
You got one more back here?
Hey, Jeremy, really wonderful talk. So I wanted to ask a question about the picks to picks, and you showed some very cool data. So my question is, have you tried it on tissue samples? We've tried it with multiplex immunofluorescence on histology slides, and we've had sort of mixed results. I wanted to see what your experience was.
I'm pretty sure people in our pathology department have, but I just work so much earlier in drug discovery. I'm usually not dealing with tissues or pathology, but certainly people are trying to predict lesions, and I don't know if they're using picks to picks or something else, but it's a hot area for safety for sure. Just not the space I'm working in.
Thank you.
All right, let's thank Jeremy one more time.